All right. Hello, everyone, and welcome. It is now 1 o'clock, so we will get started with our webinar. I am excited to welcome Meg with us today to share her research and present her presentation titled What's in Your Bucket? Understanding the Risk of AIS Introduction via Release of Live Baitfish by Anglers. I'm going to go over a couple of housekeeping items quick, and then I will pass the mic over to Meg. This is our first webinar of 2021, and it's come with a little bit of a facelift. We are going to try something new this time for Q&A, so please bear with us. But if you look on the screen now, you should see a website with an event code. If you go to that site, there's a place where you can enter your Q&A, and you'll be able to upvote other people's questions if you see something that you're particularly interested in learning about. So you can enter your questions there at any point while Meg is speaking, but we'll have that show up again once she's done, so that you don't have to worry about going in right now if you don't want to. If you're unable to access Slido, don't worry. Feel free to drop your questions into chat still, and we'll get those transferred over to Slido so that they can still get upvoted and become part of the Q&A session. We are recording today's webinar, so if you need to leave for any reason or start having some technical problems, don't worry. We'll have that up on our YouTube channel. It usually takes about a week to turn that around. And I think that's it for housekeeping. So with that, I am going to turn it over to Meg.
Thank you, Megan, and thank you everyone for attending. I'm really excited to be a part of this. I'm glad to see so many familiar, I was going to say names or faces in the room, but names in the chat, as it were. And I'm excited to be a part of this. So thank you for having me. I am going to go ahead and get set up for screen share here. And okay. Okay, how does that look, Pat or Megan?
Perfect.
Awesome, great. Well, again, thank you so much for having me. Like I said, my name is Meg, and I'm going to be talking about some of my dissertation research that was funded by the Aquatic Invasive Species Research Center. And my advisor is Dr. Nick Phelps, who is the director of the center. The title of this presentation is What's in Your Bucket? Understanding the Risk of AIS Introduction via Release of Live Baitfish by Anglers. In an era marked by globalization and a rapidly expanding trade in live animals, the spread of invasive species is a major threat to conservation. Many of the most infamous invasive species got as far as they did because they were purposefully transported by people for use as food, as pets, in gardens, or other popular uses. And in particular, infectious pathogens are really difficult invaders because they can have population level impacts on native species of concern. Humans aren't the only ones that can experience global pandemics. Wildlife can too. And a great example of this is the chytrid fungus, which has been responsible for the decline of several amphibian species and has been thought to have spread along these pathways via the trade in live amphibians. So while this is a global phenomenon, this is also taking place right here in our own backyards. In Minnesota, fishing is obviously hugely popular, and the spread of, and live bait fish are exceedingly popular live bait among Minnesota anglers. So it's important to recognize that right here in our own backyards, we have millions of individual fish being moved around the state. And several of them are being used by recreational anglers. It's a very important industry in Minnesota, valued at about $2.5 million, and supports what is a even larger industry of recreational fishing. And it's also economically and culturally important for many small towns, especially in the northern part of the state. Every year, hundreds of thousands of anglers take to the lakes and rivers, and many of them are bringing live bait fish with them. There we go. So in general, just so you could understand where these bait fish are coming from, about 60% are being harvested from the wild in shallow lakes like this one. These lakes are generally high productivity lakes, which support a very dense population of minnows. And then they're either taken directly to a retail location or maybe to a wholesaler or distributor where they're mixed with other fish. Another 40% or so of live bait fish are sourced from aquaculture. So we have both going on in the state. But it's important to note that legal bait fish have to come from within Minnesota. It's not legal to import them into the state. Once they're at the wholesaler or distributor, they might mix with other fish from other locations, and then they're taken to a retail bait shop where they're purchased by anglers. But that's far from the end of the pathway. It's actually just the beginning of another series of complex movements that these bait fish take. Once they're at the shop, they're then going to be purchased by anglers who take them to one of the over 10,000 lakes or rivers that we have here in the state, and sometimes released accidentally or intentionally into the water. Although live bait fish release is illegal, we know that it happens to some degree. So what's the concern with this pathway? Well, it's easy to imagine what could possibly happen if there was some sort of contaminant or invasive species in the bait. At the level of the shop and the source of the bait fish, we have millions of individuals being moved every year, and they're subject to minimal disease testing and biosecurity, both at the source and at the shop itself. There's also low traceability of these movements. We don't have a great way of understanding how many and when and where they're moving across the landscape. Secondly, we have millions of anglers that take to the water every year, and they are highly transient and dynamic in their behaviors. So that adds complexity to the pathway as well. In addition, we have thousands of lakes and rivers that are receiving these fishing trips every year. And like I said before, there can be a degree of accidental or deliberate release of these bait fish, along with whatever pathogens, invasive species, or other things might be present along with them. So all of this adds up to a concern about the potential for this pathway to spread AIS, and in particular, fish pathogens that might be hitching a ride along with otherwise innocuous or not dangerous fish species, including native species. Combine this with the fact that we've recently faced an increased demand for minnows, particularly certain types of shiners, and some variation in the minnow populations leading to shortages or an inability to get the shiners that the anglers actually want, means there's a lot of pressure on this situation, and we have a big tangled mess. So the purpose of this research has been to understand what the actual risk of fish pathogen spread is via the release of live bait fish by Minnesota anglers. In answering this question, it's helpful to break it down into a couple of steps. Going back to our graphic before, first, the pathogen has to be present in the live bait, whether they were sourced from the wild or raised on a farm. An angler has to purchase those infected fish and then take them to the lake and most critically, release them into the water where they can spread the pathogens along with them. And we've broken this down into 3 separate questions that I'm going to address today. First, what are the hazards of greatest concern, or what are the pathogens that we need to be most worried about? Secondly, what is the frequency of risky behaviors that allow these pathogens to spread along the pathway and eventually reach lakes and rivers where they could infect fish of concern? And finally, what is the risk of fish pathogens actually spreading to wild fish, considering what we know about number 1 and number 2? To answer this first question, we have a number of tools that we use. This shows here the results of a survey of live golden shiners purchased from bait shops across the state of Minnesota. And we found a number of non-target species along with them. All of the lots purchased had golden shiners in them, which is great, but we also found a number of non-bait species in with those golden shiners. And in addition to this, we also found a number of fish diseases, including some novel viruses that had not previously been described, as well as some viruses that are regulated state pathogens when they're found in salmonid species. So we know that non-target species and pathogens can persist in the Minnesota bait supply. But this brings us to a question of what do we actually do about it? In general, invasive species management has tended towards the precautionary principle, whereby novel or highly uncertain hazards, including pathogens, are automatically prioritized and allocated resource and risk management efforts in order to go above and beyond and just make sure that we're not getting any risk. But there's kind of a gradient of management approaches that we can take to deal with the risk of disease in bait fish. On one very far end of the risk tolerance spectrum, you could just simply decide that it's not worth trying to manage and make the decision that you are tolerant of the risk that's inherent in the pathway. Moving further along towards risk averse, you could decide to reactively manage outbreaks after they occur. Moving along, you could then decide well, maybe we want to regulate all the pathogens that we know about. And finally, at the very far side of the spectrum on risk averse, you could decide to ban live bait fish entirely and just say, no risk is worth it, and we're not going to accept any of it. So I want to just take a second. This will be the time to run our first poll, Pat. I'm just curious what you guys would like to do. So pretend you're a fish health manager and you have to decide what you want to do about the potential risk of disease spreading via live bait fish. There's not really a wrong answer here. I'm just curious to hear, based on everything you've heard so far or your experience, what would you decide to do at this point? We'll give a couple minutes, a couple seconds, I guess, to answer that. Interesting. Okay, so it looks like we're kind of split. We have a little bit of a range, maybe slightly skewed towards the more conservative risk averse side, but a lot of you are somewhere in the middle. And I'm glad to hear that, because I would propose that we try to strike a balance somewhere in the middle too. And what we decided to do in our research here is actually one of the options that's not listed here. I kind of cheated a little bit and made you guys decide. But what we wanted to do with the first part of our research here is strike a balance between the far end of allowing everything and only reactively managing outbreaks, and an extreme precautionary principle by which we would have to be responsible for testing and regulating all pathogens that we know about. So instead, we tried to strike the balance and use evidence-based hazard prioritization to identify and target the most hazardous pathogens. And this would allow us to balance the benefits of live bait use with the risks that it could be spreading invasive pathogens, allowing us to protect valuable natural resources without having overburdensome regulations. And so how we did this was via a method called hazard prioritization. What we did was we looked at the list of all fish pathogens and wanted to filter out or select against any pathogens that weren't going to be of interest for us. So anything that didn't cause disease in Minnesota fish or couldn't be transmitted by live bait fish was excluded from our consideration. This led to a list of 15 potential pathogens of concern. Once we had those 15 pathogens, we then scored them on 7 weighted criteria and ranked them according to their overall mean score. Those 7 criteria were their likelihood of transferring bait, their prevalence in the bait supply, current distribution in Minnesota, the ability for them to colonize once they had gotten to Minnesota, the host species that they infect, and their ecological and economic impacts if established. We then combined these individual scores using this weighted risk score equation and again ranked them according to those scores. And here are the results of that study published recently in Transboundary and Emerging Diseases. Along here on the lower left side of the graph here is the average risk score. So further to the right means more risk. And it's graphed against on the Y scale, the summed uncertainty score, or the degree of uncertainty that we have about those pathogens. And as you can see, we have 3 pathogens that stand out on the high end of the risk score: Asian fish tapeworm, Ovipleustophora ovirieae, and viral hemorrhagic septicemia virus. For the purposes of our study, we identified these 3 as pathogens of concern that we wanted to take and move forward through the rest of our study, as I'll describe in a few minutes. But I also want to point out that we have this sort of relationship between risk and uncertainty. And depending on where you fall on that risk tolerance spectrum, you might be more comfortable including even more pathogens for further study. Or on the contrary, you might decide, actually, I'm not worried about any of these pathogens based on your own tolerance for risk and uncertainty. So that's something important to consider. So we accomplished what we wanted to here in that we created a framework that allowed us to assess pathogens based on their characteristics and decide which ones were of most importance to us. And we applied that framework to the pathogens of interest in Minnesota and identified 3 for further study. So we accomplished this first task. Next, we had to understand what the frequency of risky behaviors was. How do we understand how these pathogens can actually move along the pathway? And to do this, we have to understand what constitutes a risky behavior. I think it helps to have a visual example of this. So when an angler decides to go on a fishing trip, there's a series of steps that occur that could result in the movement of a pathogen along the pathway. The angler has to use live bait fish. They have to use fish that are susceptible to that pathogen. They have to purchase susceptible fish that are infected with that pathogen. And then they have to release that pathogen infected fish into the water. If all of these steps occur, it's something that we are considering a risky trip, or a trip that results in the release of a fish infected with one of the pathogens that we care about. It's important to note, though, that if any one of these steps doesn't happen, it's not a risky trip. Maybe this angler doesn't use live bait, or they purchase an infected bait fish, but they don't release it into the water. Whatever it is, each of these steps has to occur in order for it to be a risky trip. So it's really important to understand the frequency with which these behaviors are occurring. In order to do this, we conducted a survey of Minnesota anglers. We surveyed via a mailed survey resident and non-resident annual license holders and received about 670 responses, the results of which I'm going to show now. So that first step in the pathway is the probability of using live bait fish. And we found that across the state, about 70% of anglers reported using live bait fish. Most of those, the vast majority of those were purchasing, but there were a few folks who harvested their own bait fish for their own personal use, which is legal in Minnesota. Additionally, we found that about 6% of anglers were reporting using live bait fish from zone 3 up here in the upper right, which is the Lake Superior watershed of Minnesota. And I'll come back to why that's important in a couple of slides. Next, we wanted to understand the probability of using susceptible fish. Each of the diseases we're interested in looking at have different levels of susceptibility for various species. For VHSV, we have fathead minnows and certain types of shiners that are susceptible. And for Ovipliostophora ovariae, we have only golden shiners susceptible to them. And it's important to understand that the the likelihood of buying a fish infected with one of those pathogens is a function of both the number of fish that they buy and the prevalence of that infection in those fish. So it was important to quantify this specifically for each pathogen of interest. Next, we had to understand the likelihood that an angler was releasing their live bait fish into the water. Thankfully, most anglers reported that they properly disposed of their live bait fish on land, but about 20% said that they released it into the water at least some of the time. And this includes anglers that harvested their own fish and then took them to a different lake and released them into that lake, as well as anglers who purchased fish from a bait shop and then took them to the water where they were released. Finally, we quantified the number of trips per angler per year to figure out how many times we needed to run the model to understand how often this is happening over a given year of fishing. So we were able to quantify the risky behaviors. We found that even though most anglers are obeying the law and disposing of their bait properly, about 20% are releasing into the water, and that these anglers have also certain frequencies of buying particular susceptible species. So we were able to quantify the frequency of risky behaviors. Now we have to put it all together. We have to understand how the actual hazards interact with these behaviors and how often risky trips are occurring in a given year. And to do this, we have to do something called stochastic risk modeling. This is going to get a little bit technical and complicated. So if this is not of interest to you, feel free to tune out for this slide. I hope you jump right back in on the next one. But if this is of interest to you, this is where I'll explain exactly how we did the stochastic risk modeling to understand what the actual risk of pathogen spreading is. So to do stochastic risk modeling, you need to start with your risk pathway. Remember, we have these series of steps, and each of them has an associated probability of occurring. When we multiply these probabilities together, we get the probability of all of those steps occurring. And remember, all of the steps have to occur in order for there to be a risky trip. But because these probability distributions have some uncertainty or variability around them, we want to make sure that we're incorporating that uncertainty into our estimates. So rather than running the model just one time with a single value for each probability, we can run the model multiple times, in this case, hundreds of thousands of times. And that allows us to actually quantify that uncertainty and variability around our estimates. So each time we run the model, there's a 1 if the step occurs and a 0 if it does not. And only if all of these across the entire row are 1 do we get a risky trip. So as you can see, most of the time we don't have risky trips. But here, excuse me, in the bottom row, all of these columns have a 1, meaning all of these steps occurred and we consider it a risky trip. So we can iterate over our model, over this calculations, hundreds of thousands of times to estimate the number of risky trips that occur in a given year and the uncertainty and variability around that estimate. So the model takes all of these inputs, these various sources of data that we got from the Angler survey, as well as historical license data, and for the scenarios that we are estimating certain hypothetical values, and then calculates these outputs. The one I'm going to focus on today is the number of risky trips occurring in a given year. And that's calculated by the model based on the probabilities that we give it for each of the circumstances. So we tested a number of scenarios for each of the pathogens of interest. For VHS, which is perhaps one of the most infamous fish pathogens out there right now, it has a wide host range and can kill a number of important Great Lakes region species. Currently, it's a regulated pathogen in Minnesota, meaning that certain species are subject to regular testing for it. And those species currently tested include emerald shiners and spot-tail shiners. However, international fish health authorities also include fathead minnows among susceptible species. So we had two sets of models that we tested for VHS. One set of models only allowed currently tested species to be tested to get the virus. And the second set of models allowed all the species, including fathead minnows, emerald shiners, and spot-tail shiners, to get VHS. So it's a wider outbreak among more fish. The second factor we modulated between these scenarios was where the outbreak was occurring. Remember before when I told you that zone 3 was going to be important? Zone 3 is important because it's considered a higher risk area for VHSV introduction. There have been VHSV positive fish found up here in Lake Superior. So if the pathogen were going to enter the state, it's a pretty good guess that it could come first into this region. So we wanted to see what would happen if an outbreak was concentrated only in that region, and then contrast that with if it was allowed to happen across the whole state. So we had two factors, two levels for each, giving us 4 different scenarios. And this is what we found. Again, this is a little bit technical, so just bear with me and try to get the high level points if you're not interested in the details, but I'll explain the details for those that are interested. Along the x-axis here is the estimate for the number of risky trips per year. So we have several million risky trips estimated for some of these. And then on the y-axis is the number of iterations, or the number of times that we ran the model and got that value. So the higher the number, the more often that value came up in the simulations. And as you can see, for the outbreak that was the most limited to zone 3, that Lake Superior watershed, only among currently tested fish, we found that about 99.8% of iterations had no risky trips occurring, or 99.8% of the times we ran the model, we got 0 risky trips. However, sometimes when we ran the model, there were some risky trips occurring, and the average number of risky trips was about 1,300. When the outbreak in the Lake Superior watershed is allowed to go into all of the susceptible species, including fathead minnows, that number jumps to about 20,000 risky trips. But as you can see, the percent of iterations with no risky trips is still pretty low, or is still pretty high, at 99.3%. That goes down to 97% when we allow the outbreak to go statewide. And when we allow a statewide outbreak among all species that are susceptible to VHS, that number drops to 88%, meaning that only 12% of trips had some sort of outbreak occurring. And the average number of risky trips in a year is over 600,000, nearly 700,000. So although these scenarios have most of the iterations occurring with known risky trips, even this most widespread one, we see that the variability and uncertainty inherent in the system means that we can't always be certain about what value is going to happen. And in fact, the average number of trips is pretty high under some of these more widespread outbreaks. We also wanted to look at a scenario model for the second pathogen of interest, which is Ovipliostophora ovariae. For those that are not familiar, this is a microsporidian parasite of golden shiners that infects the ovarian tissue of the females and causes degradation of it, including ultimately sterilization. So it has concerns for the long-standing stability of wild populations of golden shiners, as well as populations raised for aquaculture. excuse me, the scenarios we looked at for Ovipliostaphra ovariae included one under what we assume to be current conditions. We know that Ovipliostaphra ovariae does occur in the bait supply in Minnesota, so the first scenario allowed prevalence to be widely distributed and vary between 0 and 100% of mature females. The second scenario was a hypothetical one. What if we had an ability to replace 25% of the available golden shiners on the market with a clean, certified, disease-free bait source? And the third scenario is the exact same thing, except 50% of those golden shiners available on the market are replaced with a clean, disease-free bait source. The results of which are here. Under current conditions, here on the far left side in the blue, let me just say quick, this graph is the same as before with the x-axis illustrating number of risky trips and the number of iterations or model runs on the y-axis. So as you can see, under current conditions, about 96% of the time, we have an iteration or a model year, a year of fishing that has no risky trips. However, the average number of risky trips under current conditions is over 300,000, meaning that although, again, most of the time we're in pretty good shape, when we do have outbreaks in the bait supply or when we do have high prevalence in the bait supply, we have a fairly high number of introductions occurring every year. The number of, or the percent of iterations with no risky trips does go up as we increase the percentage of clean bait available in each of these scenarios, up to 97.3% and 98.3%. But as you can see, the mean number of risky trips remains high under both of these scenarios as well. So although we can decrease the likelihood, we still have an average number that's pretty high in terms of the number of trips that are resulting in the release of an infected fish for this pathogen that's already fairly widespread in the bait supply. So in conclusion, for this part of the talk, we have some good news and some bad news. I would say the good news is this, that the majority of anglers do not release their bait. The vast majority of trips, especially under certain circumstances, are not likely to be risky, and a vast majority of model iterations actually had no risky trips occurring. Under current conditions, the release of VHS positive fish is really unlikely, which is good. It means that our precautions are doing their job and keeping the risk low. However, increasing access to safe, reliable bait could continue to reduce the risk of OVO positive fish being released on the landscape. We have some bad news as well, though. Given gaps in our current management framework and the stochastic or highly variable nature of live bait fish movement in the state, the release of VHS positive and OVO positive fish is possible. And we found that when we did a sensitivity analysis of the pathway, angler release behavior was a primary driver of risk across all scenarios modeled. And so we have to understand that although we have this really great defense system, we're really only as good as our weakest link. And angler release is presenting a sort of chink in the armor against our defense of fish pathogens in the state. So we were able to figure out what the pathogens of greatest concern were. We quantified the frequency of risky behaviors, and we were able to also put it all together and quantify the actual risk of fish pathogens spreading to wild fish. So now I want to put it to you again. Based on what we talked about here, based on what you know now, I'm curious to hear the same question again. You're a fish health manager and you have to decide what you want to do about the risk of disease in live bait fish. I'm curious to see if anybody changes their answer or if it's about the same. Interesting. I don't think we budged very much at all. I'm really interested to see that. So I'm glad to see that there is a little bit of a distribution, but it seems like people's opinions did not change so much. And I think ultimately what I want to leave you with is a few impressions. First of all, I think it's really important just to recognize that this pathway and the movement of live bait fish is kind of gaining increased awareness and increased attention from particularly Minnesota DNR, but other fish health and AIS managers across the state. Because although these kinds of things can be sort of improbable, it still presents a little bit of a risk, and we need to think about how to manage it. So in conclusion, I want to leave a few points here in your minds. First, it's important to note that pathogens of concern can and do occur in live bait fish in Minnesota. Given the rate of angler release behavior and the ability for them to spread along this pathway, angler release presents a critical weakness in our defense against invasive species and fish pathogens. It's also important to recognize that we need to determine an acceptable level of risk. We need to decide if what's happening currently is okay with us. And if not, how can we reduce that risk while also preserving our important cultural and economic use of live bait? If we have to accept some amount of risk in order to preserve that, we needed to have a discussion about what that is. And finally, reducing pathogen introduction risk via behavior change, ensuring a clean bait supply that's free of disease, and increased vigilance for pathogens that are on the horizon is possible, and I believe very important. So the next project we're working on currently is another round of surveys to assess motivations for illegal bait release and try to do what we can to affect behavior change among those anglers. So with that, I would love to take questions. I hope we have plenty of time for them. I also would like to thank again my advisor, Dr. Nick Phelps, the Invasive Species Research Center, where I'm a grad student, the Environment and Natural Resources Trust Fund for funding us, and a number of collaborators here at the university, at the Minnesota DNR, AIS detectors who helped with this project, undergraduate staff who worked on this project, and members of my committee who have formed a lot of the thinking on this. So thank you to all those wonderful people. And I will turn on my video now for questions.
Excellent. Thanks so much, Meg. I am going to switch over and share my screen quick for the Q&A. So you should now have showing on your screen a QR code or a website to participate in the Q&A. So again, this is new for us. It's our first time using it. So bear with us as I learn what it's like moderating using this new tool. But if you would like to ask a question, you should be able to use your like a phone or a tablet if you'd like, and you can just scan that QR code and participate in the Q&A from your phone, or open up a website and type in slido.com and enter bait21 as your code. If you're not able to use Slido, that is okay. Go ahead and type your questions into the chat in Zoom and we will transfer them over there for you. The cool feature about this is that you'll be able to upvote people's questions. So if you see a little thumbs up next to a question that you'd really like to hear an answer to, just click that thumbs up. It'll kind of move it up in the queue and make it more likely to get answered in the time that we have. Meg did say that she's willing to provide answers in a typed format if there's any questions lingering once we run out of time. We will end at 2:00 Central Time promptly. So with that, I think I will go to our first question, which is, how does the risk of using an infected bait fish compare to releasing it? Is there still some risk even if the bait isn't released?
I get this question literally every time I give this talk. So no, the risk is not the same. The risk of using a live bait fish, in the traditional manner, simply putting it on the hook, using it, maybe it gets eaten by another fish. We understand that there is some risk of pathogen transmission, either by viral particles shedding into the water, or that fish being consumed by another fish, or maybe having incidental contact. There's some risk associated, but it's so much lower compared to the risk of a live fish entering the environment and continuing to spread that virus or other pathogen. The reason being that when a live fish is introduced, the pathogen, the parasite, the virus, the bacteria, whatever it is, can still replicate or continue to grow in that host, and it likes to be in that host. And so the number of viral particles is actually increasing as it's inside that host. Also, a fish that's swimming around, contacting other fish, maybe eating or like pooping in the water, things like that, that's going to create a lot more opportunities for transmission. The final reason that we focus on release as kind of the behavior of interest or the point of risk in the pathway is it is illegal in Minnesota. So we have this existing regulatory structure to deal with the risk, and we're trying to understand the degree of compliance with it. So short answer, yes, there is some risk associated just with use, but it's so much more when those live fish are actually released into the water.
Great, thank you. All right, our next question. Could you explain more about how pathogens are actually transferred between the fish? So does there need to be contact, or can the disease be like free floating in the water?
Yeah, it depends on the pathogen. Some pathogens have what we call intermediate hosts, where they can live in things like zooplankton, and then those zooplankton are consumed by the fish, and they continue their life cycle that way. Some of the parasites have a life cycle that includes a zooplankton host. Sometimes it can be just via contact in the water, free floating particles. Think COVID being in the same room as somebody, you can get sick with COVID. The same thing happens for fish. Sometimes it's got to be a little bit more close contact, like a social contact or mating or fighting. It totally depends on the pathogen. In general, though, they're going to be able to be spread in the water like that.
Great. Okay, and could you talk more about how you define a release? If the bait's put onto a hook and then put into the water, is that a release, or is this more of like a dumping event, or a bait
Yeah.
coming loose off the hook?
The survey explicitly asked about what people do with their leftover live bait fish. So we did not ask people how often do you have a fish escape off the hook, or a part of a fish get bitten off or something like that. We explicitly quantified the number of times people said that they release their leftover bait fish into the water. So sometimes people don't have leftover bait fish, or they didn't release live bait fish. So we defined it just as the intentional release of live fish.
Great. And could you talk more about how widespread some of these pathogens are in our wild fish populations currently?
Great question. The short answer is we have very little information about it. The different pathogens that I talked about have different regulatory structures. So some of them, VHS, for example, is on a regulated pathogens list, which means that we have fairly regular testing among the aquaculture settings. And we do have some testing of wild populations as well for that particular virus. But like I said, we only test certain species. We test emerald shiners and spot tail shiners for VHS. If a fish is not one of those species, it's not going to be tested for diseases in the same way. So we have a lot of uncertainty out there about where these pathogens are found. Ovipliostophora ovirinae, we did do a survey for back in like 2014, 2015, and it was found in 55% of bait shops, in prevalence ranging from about 10% to all of the fish there being infected. So OVO, we understand to be fairly widespread. Asian fish tapeworm was the third one I didn't talk about today because we're still working on some of the risk for that. But Asian fish tapeworm is not regulated in the state. We don't currently have survey on it. So we basically have no idea where it's distributed, if at all. Could be it's not here yet. We don't look for it, so we can't know. The long story short, though, is that we have really poor data on how widespread these are already in wild populations.
All right. So what are the methods that are recommended for anglers to dispose of live bait properly? And do you know if there tends to be more widespread agreement with using those methods amongst anglers?
So that's a great question. The second half of your question, I'll say we are going to be addressing that in our upcoming survey to try to figure out what the perceptions around disposal are. I think the official recommendation from DNR is to put it in the trash or compost somewhere that it's not going to get back into the water. But legally, you can bring it home for use later. You can throw it in the trash. You can compost it. You can do pretty much anything legally except for release it into water different than that from which it was harvested. So if you self-harvest, you can put them back into that same water, but you can't put them in a different water.
All right. Great. And what kinds of interventions do you think might be most useful in seeing some behavior change here and getting anglers to not release their bait?
Yeah, that's again something we're going to address with this next survey is kind of figure out what are the psychological and social drivers of this bait release behavior. But just some kind of preliminary information we found has said that providing anglers with an opportunity and a real tangible reminder at the boat launch or at the dock about where and how to dispose of their bait can be really helpful. If you actually provide a receptacle dedicated for bait, that can be helpful because they often feel they don't have access to a place to throw it away. There's also some work with DNR right now. I don't know if any of those folks are on the call right now, but they're doing some really cool work looking at using like stickers or reminders actually at the bait shop when you're purchasing the bait. I don't think the results of that are in yet, but I'll be watching that closely to find out, yeah, if that's effective.
All right. Great. And can some of these pathogens also be released through the water that's in that bait bucket if that water were to get released as well?
Yes, absolutely. Definitely can be, especially if they're shedding that virus while they're in transit. That's also a particular concern for aquatic invasive species, like if you've got zebra mussel veligers or other things like that. And as a result, it is not legal to move bait water between locations either. So technically, when you leave the lake, you have to drain the live well or whatever you're keeping your bait in, maybe your bait bucket, fill it with new water before you go to a new location. We didn't explicitly quantify how often that happens with this study. We just looked at the movement of fish. But yes, you have to be concerned about the water as well. And it is illegal to move water between locations.
Great. And do you have any thoughts about how to implement a pathogen-free bait supply and if something like that could be commercially viable?
Yeah, that's a great question. There's a couple of things kind of in the works right now, one of which is a program called AIS HACCP, Hazard Analysis and Critical Control Point, so HACCP. And it's currently a voluntary program that bait producers can enroll in. And it basically is training and some support to look at your pathway, look at where you're getting your bait, and at each particular point, try to reduce risk of AIS and pathogen introduction. So I think that could be expanded, maybe made mandatory so that people who get a bait dealer license or a bait producer license have to engage in these hazard reduction practices. There's also some work being done to try to secure a disease-free bait supply. The challenge with that is it's hard to get disease-free fish in the first place, and then it's difficult to keep them disease-free, especially if you don't know what diseases to be worried about. So work in progress, but as far as I know, we don't have any easy answers on that end of things. But I think AIS has it, plus trying to increase biosecurity and have a disease-free bait supply would be very valuable. Economically viable, possibly, if people are willing to pay for certified disease-free bait, then yes, that's great. It costs a lot to maintain biosecurity and have regular testing. So we have to consider that when we have regulations being added on, that results in increased burden for producers and could be difficult for some of them.
All right. Great. And I think you actually managed to answer two questions in one there because someone had specifically asked about HACCP principles as well.
There you go.
There you go. Two birds with one stone.
Yeah.
All right. The next question is there a possible way for bait dealers to inoculate the bait water tanks with something that could control the pathogens?
That is a great idea. I'm not at all an expert in actual like veterinary care of fishes. As far as I know, no. For most of these pathogens, there's not really a way, especially once they're at the bait supply or at the bait shop, for them to be treated, especially for some of these viral pathogens. For Ovipliostophora ovarii, there's some steps you can take at the level of production, if you have an aquaculture setting, to reduce transmission between different generations of fish. But as far as I know, nothing at the bait shop exists. And I don't know if it would be economically viable to do so. But that'd be great if we could.
All right. Great. And there was a question again about the containers for bait disposal, and wondering more about if the containers are not available at public landings to dispose of bait, if you're aware of any plans to improve the accessibility of those by DNR or otherwise, or if you know if like lake associations or other organizations would be able to make sure such a container was available.
Yeah, I think that would be a super effective kind of first cut at reducing release rates, not only because it provides that reminder that, signage and an actual dedicated trash can or whatever, but it also just makes it easy for people. So I think that would be a great way to do it. I do think DNR is working on some of these pilot programs across the state, one of which might include a bait disposal opportunity like that. I think it does. Um, I can maybe share with Megan some of the information about that later. But um, as far as I know, the DNR largely is not responsible for the trash cans at sites. That's often the county or um, whoever manages the launch, whether that's a private association or something like that. So more often than not, DNR is not responsible for trash cans, and they don't plan to add them to most launches. But there are some pilot programs happening right now.
Great. Yeah, and if you did have additional info on that that you want to share later, we can certainly include that in the type questions, and if there's links or something, we can have those available for people
Okay.
to follow up on that. Okay. And I think you touched on this a little bit earlier, but do you have more? Can you talk a little bit more about how pathogens ultimately get transmitted from the bait fish to wild fish?
Yeah, again, it really depends on the pathogen, and why. It also depends on which wild fish. So some pathogens, for example, VHS, they're going to cause disease in some fish, but not cause clinical disease in other fish. So a bait fish, for example, might be able to have the virus be swimming around completely unaffected or sort of minimally affected. It then gets eaten by a predator fish, and then that predator fish is infected and becomes ill. So that's one way is via actually consumption of those bait fish. However, they can also spread it to other wild bait fish. Say I dump it into a lake where there's other golden shiners or other fathead minnows living wild in the lake. They might engage in social contacts or shoaling or schooling together, and that can be enough to spread the virus too. It's important to note that this project does not quantify the risk of an outbreak occurring in the lake. All we're quantifying is the risk of pathogen introduction. So our pathway stops at the shore once those fish are released into the lake. There's a lot of factors that go into whether or not that creates an outbreak or an actual noticeable increase in disease in that lake. But we do know that the higher the number of introductions or the more infected fish are introduced, the higher the chances are that an outbreak will occur. So that's why we're interested in quantifying introduction.
Great, great, thank you. Okay. Do you know if there's any easy tests that bait shops might be able to use to test bait for some of those risky pathogens? And how frequently are bait shops reminding purchasers about proper ways to dispose of their bait?
That's a great question. The first answer about testing is no, there's not an easy, quick way. If the fish were visibly ill, that might be an opportunity for them to be removed, but they're often not visibly ill, and they might be, showing signs of stress just from being in the bait shop, like handling stress. That might not actually be a sign of a clinical infection with something. So, short answer is no. There's not a great way for them to be tested at the shop. In terms of reminders, there's been some studies done in other Great Lakes states to see how often bait shops are putting literature out and things like that. And more often than not, I think they're sort of willing and interested in it, but they don't always have the materials to do so. So that'd be another potential opportunity, would be sort of empowering and equipping bait and other angling retailers to engage on some of that communication stuff. But more often than not, I've never been told how to dispose of my live bait when I buy it. So if anybody else has, you're maybe in the minority, but it's not common.
All right. And then the next question was wondering about the response rate that you received when you sent out the mailed questionnaires to license holders, and wondering what you felt about the rate that you got and if that's enough to have a pretty accurate value for the percent of live bait released, and also wondering if you saw any geographical difference in the responses that you got.
Yeah, that's somebody that knows about survey research. Yeah, so we got a fairly low response rate. I was definitely hoping for higher. A couple of things make me feel okay about the rate of release. One is that we did an additional survey, a postcard survey that was not random. It was at sort of opportunistically sampled locations around the state. This is something that the AIS detectors actually helped with. So thank you, detectors, for this. And we found a similar rate of release when we got a little bit more spatial distribution around the state. I do think it's fair to guess, however, that this is an undercount or an underrepresentation actually of bait release behavior for a number of reasons. One being non-response bias, if people are simply not responding, and also the social desirability bias. So if people perceive that they might be in trouble if they say that they released their live bait or they don't want to report that, they're more likely to say that they didn't do it. And so I think it's a fair guess that the number is actually higher than 20%. But the nice thing is the model takes that into account because it's a stochastic model rather than deterministic. There's some fuzziness around that estimate, which allows us to kind of capture some of that uncertainty. But that's definitely a concern. In terms of geographic differences, there was no difference in the rate of release across regions, both in terms of where people went fishing as well as where they were from. So about 20% across the board released their fish. However, some regions that are really popular for fishing received more release events just because there were more people. So the rate is the same, about 20%, but more release events occur in areas that are more popular. So like cabin country, kind of in the center of the state, northern parts of the state received more releases because they're popular for fishing.
Right. Great. And so this question is wondering about other types of live bait. So, and here you've examined risks related to live bait fish. But for other types of live bait, are you aware of risks that might be around around use of those?
Yeah, definitely. Definitely. Live bait is only fish are only the beginning of the story. I think probably the one that most people are familiar with is the effect of earthworms, especially those that are not native to the area. Those introductions of earthworms have been very, very strongly correlated with popular fishing locations. So people dump their earthworms and it completely changes the forest, understory, soil composition, insect composition, all these different things. Another one is crayfish. Obviously, there's many invasive species of crayfish that we're concerned about. So releasing crayfish into the water can be a problem. Um, the release of certain types of waxworms. I didn't know this until recently, but some of those worms or little grubs develop into beetles and moths that can be of concern, especially like forest pests. So yeah, live bait is it's a whole can of worms, if you'll allow me the expression. But we focus just on bait fish and particularly pathogens because again, there's always the chance that round goby or an Asian carp or something could be present in those live bait species. But even if somebody's using legal native fish, there might be pathogens present that could be spreading along with them. So that was the focus of the study.
Okay. And do you have any thoughts or ideas about the best way to talk to people who might be opposed to disposing of their live fish and how to kind of help them understand better the risks involved and to take a less risky action?
Yeah, I think people have trouble throwing bait away for a number of reasons. Again, the future survey is really going to drill down on this. But what we found so far is people don't like to feel like they're wasting their bait. They paid good money for it. They want it to be, contributing to the ecosystem. They don't want to kill it. I think if you have a problem with killing the bait fish, I would recommend maybe not using them in the first place. It's pretty difficult and painful for them to be used as live bait. So, killing them by putting them in the trash is sort of, it's just another thing that happens when you're using live bait, I guess. If it's of a concern for you, there are ways to kind of quickly dispatch them that might be less stressful for them if that's a concern. If people are feeling like it's inconvenient, I think, again, improving the just ease of doing so with trash cans or receptacles at the launch could be good for that. You can always talk to them about the risk as well, just that there could be invasive species or pathogens present there. And you can always give them to somebody else. If you're at the lake and somebody's pulling up while you're pulling out, you can always offer for them to take over and make good use of them.
All right. Great. And let's see, do you have estimates, or do you know what the cost of monitoring for baits are? For example, if you do like eDNA tests for a particular pathogen, and if there's some logistical challenges or what those might look like?
Yeah, I'm not aware of the actual direct costs. I do know that there has been some pushback from producers on the sort of increasing number of regulations. Oftentimes, these producers have pretty diversified operations, and what you and I might imagine as like a bait fish farm is actually just a couple of tanks in the back, or a couple of ponds that they manage or something like that. So they have a lot of things going on and don't necessarily have the capital and infrastructure to invest in this huge biosecurity plan or regular testing, things like that. There is some regular testing, like I said. I'm not aware of the costs associated directly. There was a study, and I can share it with Megan later. There was a study that used eDNA to look for, I believe it was invasive carp and other invasive fish in bait shop water in Michigan, and they kind of put that forward as a more efficient way of testing for invasive species present in the water. So I can share that as well. I believe that was in Michigan, if people are interested in that. But I'm not aware directly of the costs. It can be burdensome for some of the smaller producers, especially.
Sure. Thank you. Okay. And this is a question about transfer of live bait across borders and states. So
Mm.
wondering if you find that transfer of live bait across borders is common, and is there any work on reducing that potential risk?
Yeah, great question. So just to reiterate, it is illegal to bring bait from out of state and use it in Minnesota. You can't import live bait. We did find in our study that it did happen. I think it was fewer than 10 times that people admitted to doing so, but it did happen. And we had a lot of out of state anglers report that they fish in Minnesota. Um, Ultimately, I think the frequency of that behavior is a lot higher than we know, because that's the difficulty with counting illegal behaviors, right? Is people don't admit to doing it. So it did happen, I think, fewer than 10 times in our study. It does happen, I think, fairly frequently. There have been some investigations to see kind of who might be some of the players in doing so. Uh, I think it's something that we need to keep our eyes on for sure, because even though it is illegal, without enforcement of that law, it's a gap in our armor right now. For the purposes of this study, we're making the assumption that bait was coming from within state, so we modeled the risk of within state spread.
All right. Great, thank you. So I think with that, we have just 2 minutes before we wrap up. So I think I'm gonna end the Q&A here and do our final housekeeping. So I'm gonna advance this slide here. So again, as I had mentioned, there's still a couple of lingering questions that we weren't quite able to get to, and Meg has said that she will answer those still. So when we have the recording of this ready, we will also make sure to be sending out that information as soon as we have that back. I'd like to thank all of you for joining us today for our first webinar of 2021. I hope for those of you who have participated in our previous webinars that you found this new way to interact in the Q&A session helpful. I certainly enjoyed it, and it was great to see like which questions got upvoted and to make sure that those were the ones that we were able to get to live. So as a final note, the recording will go up on our YouTube channel. I have the website showing on the screen now. You can feel free to subscribe to us as well. You should get notifications from YouTube then whenever we post new webinars or other content. You can watch your inboxes. I will send out an email through Eventbrite to let you know that that recording is available, and there will be a link to the question and answer alongside that. So it sits in the description of the video on YouTube. Finally, a big thanks to Meg for joining us and spending this time sharing this really interesting research with us. If you have further questions for Meg, I do have her email showing up on the screen as well. So feel free to reach out to her there. If you have questions for us at the AIS Detectors program about the webinar series in general, our other programs, our email is showing as well, and feel free to reach out to us at any time. But with that, I think I will wrap things up and say thanks again to Meg and to everyone who joined us. And if Meg has anything parting, I'll let you have final word.
No, thank you. I really appreciate the questions. And again, don't hesitate to reach out either via Megan or my email. And thanks again. I really appreciated being here.